Okay. So let's go through this. So these are the options for 1 through 3, and then 4 is kind of a separate multiple choice question. So out of those 4 enzymes up there that we talked about, which one kind of connects the Okazaki fragments? The ligase. Good. Which one adds nucleotides to, well, leading and technically different sections of the lagging strand? That would be polymerase 3. Good. DNA polymerase 3. Recognizes the origin of replication. Again, what's origin mean? Beginning or starting point for replication. That would be not primase, helicase. Because helicase has to unzip before primase can lay down the primer, right? So yeah, so that's actually helicase. That's the trickiest one, I think. Number 4. In DNA synthesis, DNA is, A, read from 3' to 5' and made from 5' to 3'. So remember, we're always adding on to that 3' end. So we're going to be making it 5' to 3', and the DNA that we're reading is going to be anti-parallel, so it's going to be flipped, right? I had a lot of questions and confusion as like second period, so I figured maybe it would be helpful to talk about. There's still kind of struggling with why we have to have a lagging strand in the first place. And so before we get into our new material today, which we're going to be taking some more notes today, so if you haven't gotten your notes out yet, go and do that. It's just a kind of cute little cartoon. And I think seeing the animation might kind of help. So, I do want everybody to pay attention to this. Not everybody ready to start the video? Okay, I have a time, so it's not going to start right at the beginning, but it's going to start where it starts to go through the process. So, oops. Sorry, I have my sound on the wrong speaker. Yeah.
DNA replication starts in a certain part called the origin.
Hi.
Usually this part
Hello. We started class. Thank you. Well, we were.
is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that name, keeps the DNA from supercoiling.
So we didn't talk about this enzyme, but I think we mentioned like how quickly helicase is like unwinding the DNA. That can cause some physical stress issues with the DNA coiling. So we can talk a little bit more about that.
Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next step. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons and the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of the ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these. Again, clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now,
so you can see here, as DNA polymerase is going, it has to go in the new strand 5' to 3' direction, right? It can only add on to that 3' end. So this DNA polymerase is heading this way. This DNA polymerase is heading the opposite way.
Now, the thing about DNA polymerase is when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase, once again, is building a new strand in the 5' to 3' direction. But there is a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening.
So as helicase is going in that direction towards the right of the screen, this DNA polymerase can just keep adding and adding and adding and adding and adding. That's the leading strand, right? Down here, since we have to add in the opposite direction, when more DNA opens up, we have to start over again, right? We have to go ahead a little bit and then work backwards. And so that's why we have the leading strand on the opposite side, right?
You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to name it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix deep.
So you can probably stop there. But so I was kind of hoping that animation would show you how one side can keep going and the other side, because we're getting more and more exposed DNA, we have to hop ahead and work backwards, hop ahead and work backwards. So does that kind of help a little bit more with that? Okay. All right, so I do want to
start at a certain point.
We're going to move ahead a little bit today and start talking about how we end up using DNA to make proteins. Because that's the whole reason we have to pass on DNA to our next round of cells, is so that they know how to make the right proteins. So that's what we're going to talk about today. We'll probably finish this topic on Monday, so I don't have a quick check assigned for the weekend. So, but if you haven't done it, if your study guide's not updated, it might be a good idea to use some time on that. So today we're going to talk about the general pathway of protein synthesis. We're going to talk a little bit about the differences between DNA and RNA structure. And then we're going to get into the actual process of what we call transcription. So I'll talk about what the point of transcription is and kind of some important steps in that. Okay. So we know DNA is a code. What part of the DNA actually is the code? It's well, that's replication. So that's how we copy the code, but how we read the code. If we're looking at the DNA molecule, structurally, what part of the nucleotide is going to end up coding for stuff? Yeah, the bases, right? It's the order of bases. The A, T, C, G. The order of bases holds the code. So when we did that drawing modeling activity, right? You picked your bases depending on what trait they were going to end up coding for. Obviously, way more than 3 nucleotide bases is going to be responsible for a trait, but that's the general idea. So eventually, DNA is going to give us the instructions for building proteins. The proteins are going to build our cells, and the cells are going to make up our bodies. So that's why DNA is important. It has the instructions for building the proteins. The proteins build and make up our cells. The cells make up us. So there's a really important pathway we're going to spend the next last part of this unit on, which we call central dogma. So the pathway from gene to protein is called the central dogma of biology because it's so critical and central to our understanding of the way that living things work. So, we start this process from gene to protein, obviously where's our DNA stored at? The nucleus. Oh, no. In the nucleus where our DNA is stored, part of the reason we have a nucleus is to help protect that DNA, right? To keep it safe. And so in eukaryotic cells, we do not let that DNA leave the nucleus unless we absolutely have to, which means it only leaves the nucleus when the cell divides. We need to split the DNA for 2 separate cells. So that DNA is in the nucleus, locked up tight. So how do we use that information to make proteins? We have to find a way to get that information out of the nucleus. Okay, how do you think we might get that information out of the nucleus? Yeah. We have to make a copy of it. Yep. So DNA is the original blueprint, but we don't want it to leave the nucleus. So we make something called mRNA. Specifically, there's many types of RNA. We'll get to some of those different types a little later next week. The M stands for messenger. It's called messenger RNA because it's a copy of the DNA code, right? It's a copy of the DNA message. So there's all of our mRNA copies. Generally, if we need to make a protein, we don't just copy the DNA instructions once, we copy them multiple times so we can make multiple of the same protein at the same time. That mRNA then, these are disposable copies, so they can leave the nucleus so that we can use, that process is called transcription. You know what the word transcription means? You have to write it down, place it, make a written copy. So this is what we're doing. When we're going from DNA to mRNA, we're kind of making a written copy of the DNA code in that mRNA form. So those disposable copies are going to leave the nucleus, go out to the cytoplasm, and we'll talk. There's of course a very complicated process where that code gets turned into proteins. Okay. This happens at the ribosome. You remember that cell part? Right? Ribosomes are responsible for making proteins. So they're going to use the message from the mRNA. Some other important molecules are going to come into play, and we're going to manufacture that protein. We're going to stick together a specific sequence of amino acids. And that protein is then going to play some important role in our body. It's going to cause some type of trait, multiple different effects in different traits. That process of going from mRNA to protein is called translation. Because we're translating the code from nucleotides, which has been all this point in time, right, is nucleic acid, to protein. Okay, so we're changing language here from nucleotide bases to amino acids. So that's central dogma. Sometimes replication, you'll see a little circle here for replication. I think one of the first questions on the quick check is filling in like a little diagram that goes DNA to RNA to protein. And ultimately, the proteins that you make are going to cause all of your traits. So if you look at each other and you see the differences within our human race, within all living things, those are all originally caused by differences in DNA that made different mRNA, that made different proteins that cause different traits. So again, you don't have to write this down, but this is like the Merriam-Webster definition of transcribe. So these are some different contexts outside of biology for the term transcription. Yeah.
What's the structure of mRNA?
So we'll talk about that. Yeah. So the reason we call it transcription again is because we're making a written copy of DNA in that mRNA form. So we're going from DNA to RNA, making those disposable copies. And so we're going to, first of all, see how much you can remember about the difference between RNA and DNA nucleotides and RNA and DNA polymers. Go ahead and talk about that real quick. Okay. All right. So, heard a lot of good things. One difference, first of all, they're all nucleotides, so they're still made up of nucleotide, sugar, phosphate, base. However, with RNA, we do have a different sugar. There's an extra oxygen in there, which is going to make it have slightly different properties than deoxyribose sugar. But that's the difference in the name, right? This is ribonucleic acid for ribose versus deoxyribose sugar and deoxyribonucleic acid. They also use a slightly different set of our bases. The main difference being that RNA does not use the base thymine. Instead, it uses the base uracil. But the base pairing rules are still the same surrounding that. Just instead of anywhere that we would in DNA put T, in RNA we're going to put U. So instead of T pairing with A, U pairs with A, and then C and G are still going to be the same. So those are all differences in the nucleotides. When we get to the polymer, the huge difference is that RNA is a single strand and that DNA is a double strand. Okay? DNA is a very stable, or I shouldn't say very, is more stable because of that, right? All the bases are bonded to each other. What do you think might happen to the final RNA structure if it's made as a single strand? What could potentially happen? Yeah. Yeah. Oh. Well, you have all these bases here that like to pair up with each other, right? They like to do hydrogen bonding. So instead of staying in just a straight line, what's probably going to happen to it? It's going to fold over itself, right? And so depending on what the code is, it's going to fold over itself in different ways. So we don't just have one final DNA, we don't just have one final three-dimensional structure, we have multiple different 3D structures, and so we end up having a lot of different types of RNA. Okay. Yeah.
What does it produce a separate phase just for,
I don't know if there's like a real reason why. Just over time, that's the way that the system has ended up like following that process for through evolutionary time.
Is that RNA blocked by the image? Is that SI?
Yes. Yeah. So there's way more than what's listed here. Usually in bio, especially this level, we really are only going to focus on these three. But depending on what we get to this year, we might start talking about some of those other kinds of RNAs too. But these are the three important ones we'll talk about. mRNA, again, the M stands for messenger. That's the one that carries the code from the nucleus to the ribosome. tRNA, we'll talk about that in a little bit. It helps out in the protein synthesis process, plays a really important role there. And the R, any guesses on what the R stands for? Ribosome, yeah. So ribosomes are actually made up of a lot of RNA. Yeah, Richard?
Are there like different types of DNA?
No, there's only one type of DNA structure, yeah. But many types of RNA structure. So, for example, this is a molecule of tRNA. So you can see, even though it was made as a single strand, it's folded back over itself in a certain manner. And so it's going to have a certain structure, and therefore that's going to give it a specific function, right? So mRNA is a little bit more like proteins in the fact that they're going to fold in special ways. So they have certain structures that are then going to be able to do special functions. So when we go through this process of transcription, we're going to go in a little bit more detail of the steps in just a little bit. But we have some terms here we're going to discuss. First of all, we have the DNA strand that we're actually going to transcribe. So this is the one that holds the code to make the protein. We're only using one side of the DNA molecule to make our mRNA, and that is called the template strand. Again, what's a template? What's the role of a template?
Or like, so it's kind of like a blueprint just to show, like, other copies of it.
Yeah, can you go to copies? Yeah, so this is blue. Kind of tells us where to put, what to put where. And so this is going to help act as the template for mRNA. So this is our template strand in the diagram down here that we're going to use to copy and make our mRNA. The untranscribed strand across from it, we call the coding strand. And RNA and DNA are really, really similar. So this is the coding strand is pretty much going to have the exact same code as the mRNA, except for the difference in bases, right? Instead of T, the mRNA is going to have U. But otherwise, the coding strand in the mRNA sequence is going to be identical. The transcribed DNA is the half that we're using to make the copy of the RNA. The coding strand is the one across from it that's not really involved in the process. There's actually a lot of proteins, more proteins that are involved in this even maybe than replication. But the main enzyme that you need to memorize is RNA polymerase. And so, give you a couple seconds to Work that word backwards in your group real quick. Okay. ACE tells us it's an enzyme. In this case, it's an enzyme that makes a polymer, so it's putting together RNA nucleotides, right, to make the synthesize the RNA molecule. I also want to point out, I know this might be a little challenging for some of you to see, but RNA polymerase follows some similar rules as DNA polymerase, right? So it's adding on nucleotides by connecting the new phosphate to the original 3' end of the strand. And so that's why it says here we're building it in the 5' to 3' direction. Okay. Now, when we're thinking about our DNA molecule, either side of the molecule can code for a protein. So if we're using this strand and this orientation, we'd be copying this way. If instead the code was up here, we'd be copying in this direction. Okay, so some similar directional rules apply to our transcription process. Okay, so here's a template strand of DNA. What would the coding strand be? You can talk about that in your group.
Oh, I thought.
So the coding strand is just this DNA strand across from it, right? So all the same base pair, complementary DNA base pairs. What would the copied RNA sequence be?
Literally writing down my compliment.
Okay. So that would be the complementary RNA, right? That would be made from the template strand. So you'll see the RNA and the coding strand are identical except for the change in base pairs, right? So you feel pretty okay with that before we move on? All right. So you don't have to write all this down. I'm not going to ask any questions about this, but I did just want to make you aware that there are different types of RNA polymerases, just like there's different types of DNA polymerases. And they're responsible for transcribing different types of RNA. So every type of RNA is made through transcription. We're going to really focus just on transcription of mRNA, but we make all the other types of RNA in a similar way. They're just ran by slightly different enzymes. So like RNA polymerase 1 only transcribes ribosomal RNA genes. So they're going to make the ribosomes. RNA polymerase 2 is what's going to actually transcribe our mRNA. RNA polymerase 3 only transcribes tRNA. And so we're going to talk about what a promoter is in different sequence, but they're each going to attach to different places, different DNA sequences on the chromosome. So, again, I'm not going to, you don't have to memorize what type of RNA polymerase makes what, just kind of be aware that there are different types. So, this is going to be a pretty important slide here. We're going to follow along and look at some anatomy of a basic gene. Okay, and the definition of a gene is a stretch of DNA that codes for a protein. That's one definition of it. And so what are these two black parallel lines supposed to represent, do you think? That's a DNA strand. Okay. So this little blue box here is a region of DNA called a promoter. And a lot of eukaryotic genes have this tata sequence. It's more fun to say tata, but I don't think that's the way it's supposed to be.
No, I would say.
You can say it however you want. But that's in the promoter region. What do you think promoter means in this case? If you're promoting something, so this is just really the kind of the start of the gene. So this is the start site for transcription. So again, like I mentioned, most eukaryotic promoters have the tata box. Not all. And that's going to help all of our equipment, all of our machinery for transcription to attach. Okay, so this sequence is going to be recognized by the proteins that are needed to start the transcription process. So in replication, it's the origin. In transcription, it's the promoter. So with just the promoter alone, just this sequence, we're going to be able to find the gene and we're going to be able to transcribe it. But it's not a very high level. By expression, we mean how many copies of RNA you make. So with just this promoter region alone, we can attract enough machinery to make a few copies of the DNA, but not very many. Okay, so we need another region to attract the machinery. So again, expression is the how often the information actually ends up being converted to a protein. So how much RNA we make from this, and then eventually how much protein we make from this. Okay, so that's a term you'll see a lot. We talk about gene expression, and hopefully we have time at the end of the year to kind of dig into some really cool genetic stuff. And so that term expression will probably come back then. So again, this promoter by itself isn't going to allow us to make a whole lot of copies. So we need another region, which is called the enhancer. And the enhancer is going to help attract extra proteins that are going to allow us to more efficiently go through this transcription process. We call those proteins transcription factors, okay? Because they are factors that affect transcription. So they're proteins that help out in the transcription process. Enhancers are pretty cool regions, really. So they can be located what we call upstream. So in front of the gene, they can be located downstream. Sometimes they're actually inside of the gene itself. But what you have to think about is this DNA is just a coiled mass of chromatin in the nucleus. So lots of sections of DNA are going to be overlapping and touching each other. And so the enhancer doesn't have to be right next to the promoter in order to help recruit the necessary proteins. And this is, again, kind of an extra switch. So sometimes the enhancer is going to increase the expression, so it's going to help the gene get copied more often. Sometimes it's actually going to decrease expression, so it's not going to get copied as often. Different types of cells are going to have different types of transcription factors that are going to attach to different types of enhancers. So this also helps us to only make certain proteins in certain types of cells and certain types of tissues. So even though every single cell in your body has all the same DNA, these enhancers and these transcription factors are going to help cells only make the proteins that they need to make to do their jobs. Okay, so that's a pretty important thing. Also allows for what we call inducible expression. So for example, maybe proteins only attach here if there's a certain hormone that enters the cell, so a certain cell signal or some kind of environmental signal or any kind of signal that tells our cells, hey, you need to make this protein, or hey, you need to stop making this protein. And the last slide here, when we look at the gene itself, so if we look at the DNA that's going to eventually code for the protein, there's actually two categories. We have exons, these green regions, and this is the code that's actually going to end up in the mRNA and is actually going to code for the protein. We also have introns, which are not going to code for the protein, that are just going to get spliced out. So there's a couple of things we'll talk about here though. Let me just see. You might just do a couple more things. I do my maybe take a quick break here. Okay, so before you close your notes today, just make sure you can identify each one of these parts and why it's important. Just take a couple seconds to talk about that in your group. Well, so we'll talk more about this tomorrow, but one of the things they allow for is for different pieces of DNA to end up getting attached to each other. So we can actually have one certain DNA that has so that's one possible rule. Another possible rule is just to allow for some flexibility. So there's a couple of we don't really know exactly what all those
Shows somebody going else is doing. You're like, oh. Yeah, that's much like